Neurological Disorders Lesson 3.3. Why does applying pressure relieve pain? The goals of this lesson are to introduce inhibitory synapses and to explore how inhibitory synapses modulate neuronal signaling. At the end of this lesson, students should be able to describe the flow of ions at an inhibitory synapse, describe how inhibitory synapses hyperpolarize the membrane potential of the postsynaptic cell, and explain why applying pressure relieves pain. We'll achieve these goals by modeling the pain pathway with a Socratic discussion and building neuronal circuits. To prepare for this lesson, you'll need to review the key scientific concepts that will be presented in it. They include the process of synaptic transmission, the movement of ions to create postsynaptic potentials and the effect on neuronal signaling, including excitatory postsynaptic potentials, EPSPs, and inhibitory postsynaptic potentials, IPSPs. This lesson also discusses the relationship between the different types of neuronal signals and the idea that synaptic transmission leads to the action potential, leads to more synaptic transmission, leads to another action potential, and the necessity to reach threshold before firing an action potential. You can review the scientific content in the background reading provided to you in the teacher primer, the teacher manual, and the student workbook. The teacher primer provides in-depth knowledge about the scientific content presented in the lesson. The teacher manual or lesson plan provides a minute-by-minute explanation of the lesson structure, including instructions on how to manage the discussion and activity, and the student workbook provides additional explanations for your students. You'll need to be sure to print the activity worksheet for your students. For this lesson, you'll also need white cotton balls and colored cotton balls. The key point of the lessons due now is to review the process of synaptic transmission. We'll get there by reviewing last night's homework. After giving students a few minutes to review the process of synaptic transmission with a partner, you'll use slide 3 to review the process as a class. The key point of the first activity is to elaborate on the relationship between different types of neuronal signals, specifically that synaptic transmission leads to action potentials, and action potentials then lead to synaptic transmission. We'll get to this key point by modeling the pain pathway. In the first activity, students will create 2 models of the pain pathway. The first student model has only excitatory neurons, and the students model the process of neuronal communication by passing white cotton balls, excitatory neurotransmitters, between each other. The second model introduces a special inhibitory neuron, which passes colored cotton balls and forces the other neurons, students receiving them, to receive more white or excitatory cotton balls before signaling to the next neuron. And again, the point of these models is to show that introducing an inhibitory neuron slows down neuronal signaling. The key points of the discussion are to explore the movement of ions that create postsynaptic potentials. We'll examine how the entry of positive ions creates excitatory postsynaptic potentials, called EPSPs, and how the entry of negative ions creates inhibitory postsynaptic potentials, or IPSPs. Additionally, this discussion focuses on the necessity to reach threshold before firing action potentials. EPSPs increase the likelihood of reaching threshold and firing an action potential, whereas IPSPs decrease the likelihood of reaching threshold and firing an action potential. So they are said to inhibit a neuron from signaling. We'll get to these key points with a Socratic discussion. First, to give context as to why it is important that we have inhibitory neurons, you'll ask your students, why would you ever want to decrease the activity of your neurons? The picture on slide 5 should prime students' memories of the times we experienced pain and want to continue doing whatever it is that we were doing, like playing a football game. You'll then use slide 6 to review the second part of the previous night's homework and demonstrate the molecular mechanisms underlying inhibitory synapses by asking students, how could you decrease the activity of your neurons? You're challenging them to put together 2 complicated processes, the effects of synaptic transmission and the necessity to reach threshold before firing an action potential. This is challenging for most students, but a key concept for the module, so it is important to take the time to allow students to process through these concepts, and the next set of slides allow you to further explore and develop this concept with your students. Slide 7 allows you to demonstrate how the entry of positive or negative ions changes the likelihood of reaching threshold. A note, this graph will be familiar for those classes that use Lesson 2.2 differentiated. If you chose to use the more basic version of Lesson 2.2, you'll need to be sure to give some background to this graph because students haven't seen threshold and the action potential graph like this before. See the teacher manual, primer, and student workbook for more detail and suggestions. Slide 7 is animated so you can review threshold, the voltage at which the sodium channels open and an action potential fires, and introduce EPSPs and IPSPs individually. This graph demonstrates how the entry of positive ions during an EPSP puts the membrane potential closer to threshold, making it easier for the neuron to reach threshold and fire an action potential. Thinking back to that toilet flush analogy that we used in the more basic version of Lesson 2.2, it's like making the toilet easier to flush by requiring less additional pressure to press down the lever. Additionally, the slide shows how the entry of negative ions during an IPSP pulls the membrane potential further away from threshold, which would make it harder for the neuron to reach threshold and fire an action potential. In reference to the toilet flush analogy, it's like making the toilet harder to flush by requiring more pressure to press down that lever. The last slide of the discussion connects EPSPs and IPSPs to the bigger picture of synapses. Excitatory synapses release neurotransmitters that open ion channels producing EPSPs, making it easier for the neuron to reach threshold and fire an action potential, whereas inhibitory synapses release neurotransmitters that open ion channels producing IPSPs, which make it harder for the neuron to reach threshold and fire an action potential. The key point of this activity is to provide an example of the benefit of having both excitatory and inhibitory synapses. We'll get to this key point by building a neural circuit. You'll divide students into groups and give each student a copy of the activity worksheet. This worksheet guides students through the process of building the neural circuit that allows pressure to regulate how pain is felt. Before having students complete the worksheet, introduce them to how the circuit is represented graphically by reviewing slides 10 and 11. The key point of the wrap-up is to review the pain and pressure circuit to provide an example of the benefit of having both excitatory and inhibitory synapses. We'll get to this key point by reviewing model circuits that students created in the last activity. In the wrap-up, you'll use slides 11 and 12 to review the neural circuitry that underlies the phenomenon of why applying pressure reduces the amount of pain we feel. Namely, by applying pressure, we activate a neuron that senses pressure, and that is inhibitory. And this inhibition decreases the amount of signal that is sent to the brain, so we feel less pain. The key point of the homework is to explore how our choices change our brains. We'll get there by working on unit projects. In the project, students are working independently or in small groups to research, describe, and present how different choices we make can change our brains. See the project section on our website for more information about the project and sample assignments and rubric sheets. Students often ask for more details about inhibitory synapses and how they work. You can find more in the teacher primer, the lesson manual, and the student workbook. One of the most common questions teachers get is, why doesn't applying pressure relieve all pain? And the answer lies in the fact that there is a limit to how much pain can be reduced this way, because in the end, it's a balance between excitatory synapses and inhibitory synapses. And the pain neurons are excitatory, and if you're in a lot of pain, that's a lot of excitation that you would need to inhibit with pressure. At the end of this lesson, students should appreciate the significance of both excitatory and inhibitory synapses, including what ions flow into a neuron to generate an EPSP or an IPSP, and how that process changes the membrane potential to either bringing the neuron closer or farther away from threshold, and the likelihood of firing an action potential. This lesson builds on the previous lesson's discussion of the process of synaptic transmission. This lesson also links to the previous unit's discussion of action potential and threshold. And finally, this lesson lays the groundwork for understanding pain phenomenon and neural circuits, which we'll be discussing in future lessons. Don't forget, if you have any questions, concerns, or feedback, to let us know. You can contact any of the CTSE team members, and we'd be more than happy to help you.